Hello, my name is Narasimhan Balakrishnan, and I'm a doctoral candidate at Northwestern University. Today, I'm happy to be talking to you about some work I've done on model reduction and control for multicellular biological oscillators. So the oscillator system of our interest is the master or core circadian clock, which is both high dimensional as well as spanning multiple scales. So at its core, this core circadian clock is a network of specialized neurons, and each neuron or cell in this network has a gene regulatory network, which you can see kind of from this wiring diagram, has a few species and has a fairly high dimensional, or rather has fairly high dimension in itself. So you can see that this system spans multiple scales and has a lot of species that are of interest. And the other part of the motivation for this work is circadian phase adjustment, which is essential to fixing things such as jet lag, so on and so forth, as a problem that we might treat in a classical control manner. So say we had one of those species of our interest in this system that was oscillating, and we wanted those oscillations to be advanced by a certain amount of time or by a phase angle. And we know, for example, the circadian system can be adjusted using exogenous inputs such as light or small molecules. We all take melatonin to hopefully help us sleep. And we could treat this similar to how we might treat a classical control system where we have an input, a dynamical system, and we have certain outputs that we're interested in. So we could then also choose to describe this mathematically with a system of differential equations and come up with a cost function for the control problem we're interested in. And the control problem we're interested in here is that of minimum time. So this could be, for example, say I'm flying from the East Coast to the West Coast in the United States and I want to get over jet lag as soon as possible. This might be the kind of problem I'm interested in. And this is where both those tie into the motivation for some of the model reduction work that we do, is because, as I mentioned before, this system is fairly high dimensional. So in a system that spans anywhere from tens to thousands of cells, the dimension is 10 times that. And for a cost function of the form written below, rather written previously, it's rather infeasible to optimize it in reasonable time treating the system with those kind of equations. So the approach we use is that of reduced phase models. And the idea behind a reduced phase model is fairly simple in that if we have a limit cycle, or we don't look at the mathematical description of the high dimensional limit cycle, but more of an angle or phase angle with respect to a certain phase. Think of it as if you have a point moving around in a circle, we might be less interested in the absolute coordinates of the point, but maybe more interested in the angle that point makes with a certain reference as time progresses. So we go from our large dimensional ODE description of the system to one that just describes the individual phase angles in terms of the individual natural frequencies, the response of the system to small perturbations, which is called the impulse phase response curve. And because this is a system of coupled cells, we also have a coupling term as well as white noise to account for sort of measurement errors, so on and so forth. So with this reduced model, we also go one step further to write a method of moments kind of equation with order parameters in sort of a self-consistent form and use that self-consistent equation to infer the parameters of this reduced model using a Markov chain Monte Carlo scheme. And the data that's used to train that is simulation data from simulating the full high dimensional model under different conditions with different perturbations. So some of the results we get from making these reduced order models, we can see that if I were to plot the individual time periods in a population of 50 cells of the full high dimensional model and plotted the reduced model, the time periods of cells from the reduced model inferred from the same, we see they're fairly well correlated. In fact, the R-square value across all of the reduced models that we infer for populations of different sizes and heterogeneities is always more than 0.9. We can also see that when we compare the phase response curves or the perturbation data, that the mean squared error is often less than 5 to 6% of the full simulations. But we do know that the control problem is not as straightforward as the open loop diagram that I showed a few slides ago. So we need to actually put this into a proper closed loop where we integrate both the reduced phase model we have in addition to the full ODE model. So what the controller does is optimize a much smaller functional and then feeds the optimal input into the full model. And preliminary control simulation results, the problem I'm going to talk about, this is one of the same minimum time where we want to shift someone's phase by 4 hours. Say they're flying from India to England or from California to Hawaii. And the input we have is pulses of light that are 5 or 10 minutes long. And this is a constraint because these are the inputs that were fed into full simulations that were used to identify the reduced model. And what we do see is that this fairly significant phase shift can be achieved, or that phase can be adjusted in roughly 4 days or so, even at high heterogeneity levels in the population. So to conclude, we have this reduced order modeling framework for this high dimensional system, and our reduced model recapitulates natural features of the system such as biological variability. It recreates, or rather recapitulates, the full model sensitivity to perturbations, and we integrated this reduced order model into a closed control loop with the full model. So in future work, we want to improve this reduced model by adding a wider range of inputs to the full simulation, as well as extend our control simulations to larger systems, different systems, to make inferences regarding the effect of biological variability on how easy or difficult it is to control the system. With that, I would like to acknowledge my lab funding sources and thank you for your attention, and I'm happy to take any questions.